So I'm going to call up our next speaker, another esteemed friend and colleague, Camillo Ricordi, at the Diabetes Research Institute at the University of Miami. Perhaps one of the leading stem cell, if not the leading stem cell diabetes researcher on the planet Earth. Camillo is one of my original, the Genetics Policy Institute original scientific advisory board with such luminaries as Doug Melton, Dolly the Sheep's father, Ian Wilmot, Rudolf Janisch, and other giants of the field, and was at the first meeting that we organized, which was for the United Nations, on human cloning in all its aspects for the United Nations back in 2004. So that background as being an alumni of that meeting, which was a game changer and steered the United Nations from banning a form of stem cell research worldwide by treaty, was very significant to me. There's no one that has helped me more as an advisor and learning about the science of stem cells than Camillo Ricordi, and I'm very pleased to introduce him to give a presentation at the World Stem Cell Summit. Thank you.
Thank you. Thank you, Bernie. I'll try to give a little an overview of how our thoughts is evolving in cell therapy for diabetes and beyond diabetes, because the applications are much wider than diabetes itself. So these are my disclosures. Since I have only half an hour, I cannot go through all of them. But the one relevant for this talk is that I'm a co-founder of ION Healthspan and board member with my co-worker, collaborator, Chief Scientific Officer Giacomo Lanzoni, who you met already. And the other disclosure is that I published the Bible of Healthspan recently. In Italy, it was a bestseller, but Mondadori called me that I was beaten trashed by Prince Harry Spare that jumped to the first place. So I'm sorry, I'm no longer, but I will still be in the Oscar bestseller permanent collection. And I did the translation in English. I don't want to sell books. The revenues are going to the Cure Alliance to support Healthspan research. But there is too many concepts in 280 pages that I will not have time to run randomized trial in the next 30 years to prove everything that I think. So I'm doing a little like a small scale of David Sinclair concepts. So my story, I've been involved in cell therapy and cell processing all my professional life, from the development of the method to isolate islets from the human pancreas and all mammals' pancreas, to the first demonstration in animals, the first reversal of diabetes in mice with human islets obtained with that method, then the first successful allotransplant at the University of Pittsburgh with Thomas Starzl when we formed the journal Cell Transplantation, and then the Cell Transplant Society, and then all the way to the performance, the successful completion of the phase 3 trial of islet transplantation to treat the most severe form of diabetes. And manufacturing, cGMP, advanced cell and biologic manufacturing remains the center, the heart of our institute in Miami. And we have this program that is called Fast Track Center for Testing, that is to offer industry and academic center a way to test their technology without interfering with intellectual property, from in vitro, small animal, non-human primate, and pilot clinical trial if necessary. So these are some of the milestones that I don't have the time to go through all of them, but I just want to point out that some of the most exciting ones have been very recently, including saving patients with COVID-19 during the pandemic with umbilical cord MSCs infusion, and then the first in-human successful transplant of stem cell-derived islets, and now upcoming trial, one was just approved, allowed by FDA, that is the Vertex VX264 with stem cell-derived islets without immunosuppression, and an upcoming trial in a year where cGMP manufacture is in progress to hopefully perform another transplant of stem cell-derived islets without immunosuppression using a fast ligand and rapamycin transient immunomodulation. Islet transplantation has been moved forward in all the world except the United States because we've been blocked by FDA outdated regulations that were established 2 decades ago or more. So we are still considered and regulated as advanced cell therapy, while Europe, Canada, Australia, Japan, everywhere else, they're regulated as organ transplant because they're not cells, they're mini organ. And, but I don't have time to enter in this debate, but I'm happy that most of my students and fellows and now leaders of other institutes worldwide are moving forward. For example, I'll mention in a minute. The new breakthrough that really caught our interest was that the Vertex trial announced the first patient off insulin with a stem cell-derived islet transplant. I'm honored to be PI of the Miami site and chairing the steering committee for the Vertex, both trial, the VX-880 that is in the liver with immunosuppression and the new one that will be a macro device vascularizing implanted without immunosuppression. We presented the data in Stockholm at the ESD and at this other webinar, but practically it was quite impressive, and I'm not at freedom to discuss the results of the other patients, but will be presented at ADA in San Diego at the end of this month. So stay tuned for that update. This is the technology that Doug Melton, Felicia Pagliuca, and Sema Therapeutic originally developed and now is in clinical trial. What has been quite spectacular is that the timing range was like 99.9% timing range with patient off insulin with an hemoglobin A1C that went from 8.6 to 5.2 on no exogenous insulin. So it was very exciting as first successful stem cell-derived islet transplant. The field of islet transplantation, though, is teaching us something that we have to be aware of, because from the first in-human success in stem cell-derived islet transplant to a cure for all patients with diabetes, we have several challenges, requirement, possible solution, but the challenges we are learning from the islet transplant field, because if you see the results that were recently published of the famous Edmonton trial that was initially very successful, you see that over 20 years, the percent of insulin independence is down to 8%, but graft function is still 48, is almost 50% graft function, which is very important for patient benefit and survival. But still there is this attrition of the cell therapy. And there are many reasons for this, the glucotoxicity, lack of innervation, immune suppressive drugs that are very bad for islet survival because they are diabetogenic. You can induce diabetes in a non-diabetic subject receiving a kidney transplant just using these immunosuppressive drugs. So you're actually using something that increases the metabolic demand of the transplanted cells 2.5-fold. So you're already at the time of transplant with a marginal mass, if you wish. Nevertheless, the improvement have been going on in Europe, in the countries that were allowed to move forward in the field, while we are blocked in the United States by FDA. And here is France, a single center, François Pateau and Julie Carconte in Lille. They performed 30 transplants since the beginning of the year. That is more than what we did in the United States in the last 5 years, all over the country. And with spectacular results, they improved the graft function and survival at 10 years to over 75–80%, as you see on the left side. And France approved islet transplantation as standard of care, fully reimbursed by the system, by healthcare system. So France, like Canada, Italy, other places, Australia, UK, can now move forward and live as in the dust, thanks to our regulations that are impeding us to move forward. So What can we do now to move to eradication of a disease beyond an initial cure? Now we can treat diabetes, you can reverse it, you can have graft function for 20 years in 50% of the subjects, but we now have to concentrate, and is one of the reasons of the Healthspan code that links like long-term success in diabetes cell therapy to long-term success in preventing accelerated aging associated with diabetes and other disease conditions on inflammatory base. So we have to prevent disease recurrence, prevent accelerated aging and its associated chronic complication. Despite immunosuppression, we analyzed our data in the last 20 years, and patient survival has been spectacular following islet transplantation. It is actually superior, even if it's not a randomized trial that will never be able to be afforded by anyone, but is 85% cumulative survival at 20 years, while in diabetes with insulin in the same age group, because diabetes, what we have is we have this accelerated aging, but it starts after age 40. You see this slope. Everything is okay up to age 30, and then suddenly you have this phase of decline that we all experience also in non-diabetic population, but is a chronic accelerated aging, so this phase of decline increase after a certain decade. And the results now will be better with modern technology, artificial pancreas, but is still characterized by accelerated aging. And islet transplantation to the right, you see that can have an impact. So if you are in an age range of 40, 45 years of age, immunosuppression may still be better than just insulin without immunosuppression. And the accelerated aging in diabetes as invisible killer has been outlined very well in this paper in The Lancet, showing that the mortality of people with diabetes type 1, if you're diagnosed before age 10, you may lose up to 2 decades of lifespan. So that's why we have progeria as an acute model of accelerated aging. You can use diabetes as a chronic model of accelerated aging. And why we are so focusing now on health span instead of lifespan? You know, longevity now, a lot of countries benefit more than 80 years of lifespan expectancy, but that has been sick longevity, unhealthy longevity. And the phase of decline affects, if you see, we are the prototype in the United States of what not to do for healthy lifespan. If you see the top is Japan, number 1. Italy, we are not doing that bad. We are in the top 10. But still we would be excluded for the soccer World Cup in that ranking. And then you have United Kingdom is 28, and United States 69, together with the Arab Emirates, and Saudi Arabia is lower, with 66 years of healthy lifespan. And what we say that 90% of Americans after age 65 are affected by at least one chronic disease condition that is largely preventable, which is also responsible for 90% of the healthcare budget that now reach $4.3 trillion or 20% of our GDP. So if you think that this is an age group that will double in the next 2 decades, this is becoming also economically unsustainable besides the suffering. So interestingly, if you look, what is a predictor of accelerated aging and shorter health span is insulin resistance. You can look at telomere, biologic aging marker, or the epigenetic modification, but the simplest thing is to do a simple test, do a home insulin resistance, check your fasting glucose, fasting insulin, your AAPA inflammation ratio, your level of vitamin D, and you can have a very simple way to see how good or bad you're doing with your diet and lifestyle and everything else. And is a major factor accelerating human healthy aging. And the consequences are not just diabetes, but kidney disease, hepatic disease, risk of cancer, Alzheimer, neurodegenerative, arteriosclerosis, and accelerated aging in general. So now if you apply this concept to what do we need to do to move from the first in human success to a cure for all patients with diabetes and beyond diabetes, Of course, we need to improve the transplant side. We need to induce tolerance, avoiding anti-rejection drug, and we need an unlimited supply of insulin-producing cells. So we published a few years ago a new way to bioengineer an intra-abdominal endocrine pancreas with a biologic scaffold that allow you to recreate an endocrine pancreas in the abdominal cavity. This was the first success in an extrahepatic site for an islet transplant. And it's a very simple technique. You just drop coins of islet and autologous plasma with human recombinant thrombin to create this biologic scaffold, fully resorbable once the islets are revascularized, that produce long-term insulin independence. And now we are combining this with technology to induce tolerance, as I show you in a minute. So now that we have the first trial without immunosuppression, the Vertex 264 that has been recently announced and allowed by FDA, we may have down the line the possibility to have unlimited islet supply from stem cell-derived sources and no anti-rejection drugs. That will be a must, because if you need chronic immunosuppression, the indications will remain limited to the most severe cases of diabetes, and then you don't need stem cell-derived islets. You can just do it with a few thousand organ-derived, multi-organ donors-derived islets. Mission possible, but not mission complete. There are still several challenges. We'll have to learn a lot and hopefully learn from the history of islet transplantation, that we use it as a model of cell therapy for diabetes. Another interesting development is the clinical trial now of anti-CD40 ligand co-stimulatory blockade that is replacing Prograf, the calcineurin inhibitor that is the most toxic immunosuppressive agent in islet transplantation, and just replacing Prograf with anti-CD40 ligand. We show in non-human primate that you can triple the level of C-peptide, of insulin secreted by the transplanted cells, so very promising. We're really looking forward to start this trial. Also in islet transplantation is already ongoing in kidney transplants. We have eye-tolerance that is now combining the biologic scaffold that we developed and prove clinically viable with this fast ligand microgel that induce apoptosis of the infiltrating effector cells and increase regulatory T cells that are maintaining long-term tolerance. And this is a very exciting new development moving towards clinical trial, hopefully in 1 year, with the cGMP manufacturing already ongoing after it was shown to work already in non-human primates. So very promising technology. So now for long-term success, avoid insulin resistance, disease recurrence, and accelerated aging, we are looking a lot into mesenchymal stromal cell or medicinal signaling cells, or what, to avoid debate on how to call them, just MSC, Tregs, and Healthspan strategies. So MSC, we all know, we discussed where they come from. We are focusing on perinatal sources of MSCs, like umbilical cord, especially derived MSC, because they can be of baby origin at time 0. It's the youngest cells you can get without going to embryonic or fetal cells. We have a long history of working on MSC, first in organ transplantation, then in diabetes, and most recently in COVID-19. And now we're starting a trial in diabetic nephropathy. And we have one trial approved for blocking autoimmunity at the onset of type 1 diabetes with this technology with perinatal cells that will be injected directly in the pancreatic arteries to try to block autoimmunity within the first 6 months from diagnosis. And this is a trial that has just been approved recently and is moving forward now. Hopefully will start very soon. The real surprise and happy coincidence in the disaster of the pandemic was that we were blocking the diabetes trial, but we had ready a master cell bank and therapeutic doses in the DRI cGMP, and we were able to obtain a very fast, it's the fastest FDA approval allowance of an IND I ever got. Like in a week we were approved to move forward. And we showed that this, in green, the people receiving this infusion, this was a double-blind, placebo-controlled nobody, the patient or the physician, didn't know what they were receiving. Of course, the patient was easier because they were intubated in the ICU, dying, and in that time, the mortality in the control group was still very high, was over 50%, while we were able to save all the people in the stem cell treated group. And what is important, besides saving all these subjects, was that we proved the immunomodulatory anti-inflammatory effect of these cells that was incredibly powerful. So we could block the cytokine storm and have a powerful immunomodulatory effect, which is now confirming mechanistic studies and at the base of a potency test that we developed for phase 3 trial, so that now we have a technology to compare stem cell product or MSC products, not stem cell, for their potency and potential efficacy. So this study confirmed a powerful anti-inflammatory immunomodulatory effect and was approved for phase 3 trial, and now we do scale-up manufacturing, 3D bioreactor, and besides this, MSC can promote tissue repair and regeneration, so now the application in clinic will be very much expanded. One of the most important findings that Giacomo Lanzoni, who is here, who was the principal investigator also of the COVID trial, is that there is a strong effect on activated Tregs. You have more than 100% increase of regulatory T cells. So if you think of MSC to potentiate any strategy that wants to increase regulatory T cells, from the trials in Poland of Tregs to the eye tolerance trial to other, it could be a very nice combination therapy to induce tolerance. So now, prevention of accelerated aging and associated chronic complication. We have published recently that using this cell in long-term diabetes, you can prevent the development of neuropathy, nephropathy, and retinopathy. That are the three most feared complications of long-term diabetes associated with the accelerated aging that you have in the disease. So this has encouraged us, say, if we can prevent this complication, what about blocking and reversing them at the beginning when you have them associated with accelerated aging because of our unhealthy lifespan, even in the absence of diabetes. And this, I don't have time in the next 4 hours to tell you everything about this book, but the pillars of this concept are integrating physical exercise, healthy nutrition, protective molecules, and with MSC-based intervention as needed. And of course, as we discussed, in a healthy planet, because we can do it's not worth to increase our healthy lifespan if we lose the planet where we live, but that's another problem. So the problem is we have 100 autoimmune diseases responsible affecting 20% of the population. Over age 65, 90% of people in the United States suffer at least one chronic disease. This disease accounts for 90% of the 4.3 trillion, I need to update these slides, and is a population segment that will double in the next 20 years. So that for the first time in history, lifespan has decreased in 3 consecutive years in the United States, and is now thought that children born today may represent the first generation living less than the generation of their parents. So the goal is not simply to live longer, but to live longer in physical and mental health, compressing this phase of decline that you see on the left, aiming at this famous square wave, so that you arrive healthy at whatever age you want to arrive, run a marathon or do your favorite physical activity, that for me is not a marathon, go to sleep and don't wake up. That would be the perfect. So we are focusing on MSC, protective molecules like vitamin D, omega-3, polyphenols, SIR2 inactivators, to try to counteract this perfect disease predisposing storm that we've been able to develop because our trends in nutrition, increased refined carbohydrates, omega-6, processed foods, sedentary lifestyle, while increasing then the protective factor that decrease, like omega-3, polyphenols, SIR2 inactivator, vitamin D, and physical activity. So why omega-3? It's been shown already that omega-3 can stop progression of autoimmunity in type 1 diabetes, but more importantly, if you have omega-3, you can double the Tregs and decrease the Th17, that is aggressive cells. And if you have instead arachidonic acid as an omega-6 surrogate marker, your Tregs goes down to 0 on the panel to the right, and your Th17 goes through the roof. So you find yourself that a simple lifestyle and dietary situation can impair your ability to fight a triggering event of autoimmune diseases, responsible of the first pandemic of autoimmune diseases affecting 20%. Or if you have SARS-CoV-2, you're unable to modulate immunity and inflammation and get into the cytokine storm that can give you problem in that direction. So it has been shown that if you have low levels of omega-3, you have 4-fold higher risk of developing diabetes if you are antibody positive. If you have vitamin D and omega-3, you can have almost 40% reduction in all autoimmune diseases. This was a very large study, vital study, funded by NIH. This is an incredible data from Israel that show that if you have vitamin D low level deficiency, you have not twice, not 4 times, but you have 14 times more probability to develop severe COVID or die from COVID. So for those like the Italian authority that says you don't need vitamin D above 20, like think again, and before you follow indication from any large organization, from the one that starts with a W to others, like think twice and read more about what is happening in the literature. So this is a paper we did recently on protective factor to halt or mitigate autoimmunity, viral pandemic and other diseases. It was interesting that this combination of polyphenol and sirtuin activators induce a 99% inhibition of viral replication, not just of SARS-CoV-2, but also of influenza type A virus. We have data on sirtuin and aging and brain that, of course, sirtuin still need to be confirmed in large trial, but there is more and more evidence emerging and indication that may have a role from Alzheimer, from neurodegenerative disease. We just recently published in a model of Parkinson's disease that they can decrease inflammation and pro-inflammatory cytokines, decrease reactive oxygen species and apoptotic mechanism while increasing cell differentiation into the dopaminergic neurons. There are even indications for the treatment of diabetes, and we are now start testing in diabetes combination of vitamin D, omega-3, and a combination of polyphenols and sirtuin activator that is giving initial very promising result, but they are anecdotal. I not even want to report them here, but we need funding hopefully to do randomized controlled trial. Problem is that if you are using generic drugs, like the supply of vitamin D cost $20 for 3 years. And when I went to my friend, head of a big pharma, asking, do you want to fund this trial for 5 to 10 million? And he says, excuse me, $20 every 3 years? Now I'm working on molecules that are a little more interesting on a business perspective. But insurance should be interested, healthcare system should be interested, we should all be interested. So I don't know if I will ever be able to do randomized trial with thousands of people, but I said at least I write everything in a book and share it, helping one person at the time while we try to develop a cure for all. So in summary, we have a lot of room for improvement, if you wish, in United States as far as healthy life expectancy, but we have way that we could do it, and this is not only to prevent suffering, but the economic value of targeting aging is becoming evident. We used to say, who can afford to live until 120 years, but is not living longer, is living healthier and arriving to the end in physical and mental health. So recently, David Sinclair and others published this with Oxford and UK and Harvard, that every healthy lifespan you add, you can save 38 trillions to the global economy. So 10 years could generate 367 trillion in saving that could be used to fight global warming, hunger, or any other challenge that affect us in this century. So I thank you for your attention. I thank Giacomo Lanzoni, who has been my left and right arm, Matthias von Herrat, who joined us in Miami as director of the Institute, the whole team, regulatory team, Shuming Su for the advanced cell manufacturing, and all our collaborators in the US and worldwide. Thank you very much.